Closely related to taste is olfaction, or the sense of smell. We'll now move on to discuss olfactory receptors, as well as the smell receptor pathway. The olfactory system. This image depicts the layers beginning at the nasal cavity at the bottom of the image. First, the olfactory cilia. This overlies the olfactory epithelium. The next layer is the lamina propria and the cribriform plate, as well as the olfactory nerve fibers, which eventually synapse with the olfactory bulb and become the olfactory tract. The olfactory system is part of the chemosensory system, along with taste, that detects chemical compounds. The human sense of olfaction or smell is based on olfactory receptors detecting odorants which are dissolved in the overlying mucous membrane. Odorants are introduced to the olfactory system through normal breathing, eating, and sniffing. Like the taste receptors, olfactory chemoreceptors are replaced approximately every 60 days. There are at least 6 primary odor qualities: floral, ethereal, musky, camphor, putrid, and pungent. These are detected by olfactory receptors, and there are at least 1,000 different receptor proteins, each sensitive to a different specific odor. The structure of smell receptors. Olfactory chemoreceptor cells are bipolar cells. At the apical surface are the cilia that detect the odorant. At the basal surface, an axon extends into the olfactory nerve filaments. Specific odor-binding proteins in the nasal cavity bind to a specific receptor on the cilia. Once the specific odor-binding protein in the nasal cavity binds to a specific receptor on the cilia, this leads to the activation of olfactory nerve filaments. These filaments pass through the cribriform plate into the olfactory bulb. There, they group and form synapses with the dendrites of tufted cells. The axons of tufted cells leave the olfactory bulb and enter the olfactory tract, where they eventually project to the olfactory nucleus and other areas involved in smell in the cerebrum.